So, hi and welcome everyone. Before we actually go into the presentation, just a very brief disclaimer. Jeroen and I are both working at a company that is called Resolve Biosciences and that is pioneering spatial transcriptomic technologies. That said, this presentation will not be about the work that we do at Resolve, but is really meant to provide an overview over the great technological advances that enable us to study spatial transcriptomics or spatial distribution of gene expression today. And to lead you into the topic, we thought it would be worthwhile to give you a brief introduction into the past and current methods used to study gene expression. So let's go. Now, early approaches, and for me, these are all the methods used before the advent of next generation sequencing, can be sorted into two larger categories, untargeted and targeted methods. Now, the goal of these untargeted methods was to capture and sequence short stretches of transcripts and then link that information back to the gene sequences in order to find out what genes have been expressed. And even though this is a very similar approach to what is done in RNA-seq today, the biggest limitation of this technology was that sequencing prior to NGS was super slow and super expensive. And of course, the genome annotations were suboptimal. So this technology had some major challenges that it needed to overcome. The second category, targeted approaches, are based on the hybridization of probes that have a complementary sequence to the transcripts that are transcripts of the genes of interest. And these targeted methods, such as RNA microarray or RT-PCRs, are generally used to confirm the absence or presence of certain genes in a tissue or cell. But they can also be used to monitor relative changes in gene expression between two experimental conditions. Now, the major limitation of these targeted technologies is, of course, that they do require some prior sequence knowledge of the genes that you're interested in, which can make it difficult to work with species for which no sequencing information is available. So today, the predominant method in the field is RNA sequencing, which is basically a high throughput approach to untargeted gene expression analysis that became possible through the advent of NGS. So in RNA-seq, scientists homogenized pieces of tissue, captured the majority of the transcripts from the lysates, and then sequenced them, providing some detailed information of what genes were expressed in the investigated tissue. And of course, the increasingly improved genome annotations were a big help in the process of identifying genes. And then single-cell technologies came along, such as drop-seq or indrop, that were fostering the single-cell revolution. Because these technologies now allowed the barcoding of thousands of individual single cells. So now, instead of receiving tissue scale information of the transcriptome, as we have just seen in bulk sequencing, it was now possible to obtain the transcriptomes of thousands of single cells at the same time, providing us information on the heterogeneity of transcription within tissues, which was, of course, a great next step. However, even though RNA sequencing today is the predominant method in transcriptomics, it also poses a big challenge to the community that we want to talk about today. And we call this challenge the spatial gap. The problem is that even during the process of tissue dissociation, where we are looking at single-cell resolution, all spatial information of the tissue is lost, and we cannot trace back what cell has been located where in the tissue prior to the dissociation process. And that means that after obtaining a lot of individual expression profiles in a single-cell RNA-seq experiment, we cannot put the pieces back together and reassemble the tissue. And this is a problem because we know that the position of a cell within a tissue is crucial for its function and the functioning of the tissue itself, because context matters. And this is where spatial transcriptomics come in. And what I mean by this is that these methods now allow us to link the data that we have on the transcriptome of individual cells to their actual position within a tissue, or even better, to obtain detailed information on the transcriptome of a tissue while maintaining the spatial information in the first place. Now, of course, the fact that context matters is nothing new, and thus it is not surprising that earlier methods have tried to preserve the positional information of individual cells within a tissue. And here again, two avenues come to mind. Either one tried to carefully isolate cells with a known, from a known location within an intact tissue, as it is done in laser capture microscopy, or one keeps the tissue intact altogether and attempts to locate transcripts within their native location, as done in RNA FISH experiments. However, the main challenges of all these techniques, and these are just two for reasons of display, is their very low throughput and the low capability to multiplex. So these methods could not really keep up with the thousands of transcripts that were isolated and identified using RNA-seq approaches. So in the next section of this presentation, Jeroen will now provide you an overview of different technological approaches that we use today that link the spatial information to transcription profiles.
Thank you, Jasper. So indeed, the current state of the spatial transcriptomic landscape has many technologies that allow you to really go beyond what we know from traditional single molecule FISH in terms of multiplexing. So there are many technologies allowing you to interrogate a lot of genes per sample, and you can categorize them in 4 major different classes. So I will go over each of these classes and really explain you the strengths but also the weaknesses, and these classes are really defined in how the chemistry works. So the first class is called cyclical FISH. So it's based on single molecule FISH, but instead of just interrogating 3 or 4 individual genes per sample, what you do is you image those, but then you strip away the probes and you rehybridize a new set of genes in a next imaging cycle. And you actually repeat this process, and this was first demonstrated by OSM FISH. So you repeat the process until you have enough genes per sample. So this allows you to really go for higher multiplexing. So the resolution and detection sensitivity is really on par with what we know from single molecule FISH. However, the multiplexing capacity is still relatively limited. Because you strip away these probes, it actually has an impact on the RNA quality over time, because you're rehybridizing, re-imaging, stripping away these probes every time. So we see a drop in signal intensity the more cycles you want to add to this protocol. So the way people try to circumvent this is really going into barcoded FISH. And in barcoded FISH, you really probe the same target over and over again, but with different sets of fluorophores. And this is really allowing you to generate barcodes up front in the way you design the probes and which fluorophores to attach them to. And so how it actually works is, for example, in this example for transcripts originating from gene B, we know upfront in the first round we expect them to have a red signal, and in the second round a green signal. And at the very end of all these individual rounds, you can basically take all these individual fluorescent readouts and consider them as a barcode. You decode that barcode and you can assign the identity of those transcripts to that gene. So it's a very powerful method. And you can think of this really also as a scalable method in which you have the number of fluorophores to the power of the number of rounds of imaging. And as you can see, it very much scales to, let's say, about the whole transcriptome that you can image in a single sample. However, it's not that straightforward. There are severe limitations and many reasons why we cannot obtain these type of numbers here. A landmark technology using this chemistry is called SecFISH, developed by Long Kai. And basically what you do is, as in the example I gave earlier, you target a number of RNAs and you remove these probes in between, and you just label them again with a new set of probes with a different fluorophore. And this allows you to really build that barcode and decode it at the very end. You also have some variations where you increase the signal intensity by amplification methods. And this is showing you some images of how it actually looks. So the resolution is really single molecule FISH again, very good, and you can actually do massive multiplexing up to 1,000 or even more genes per sample, so very powerful. but the efficiency is not always on par with what we're used to from single molecule fish, and this has several reasons. So one of the reasons is that there's no error correction built in. So for example, if you have a mishybridization event, you actually lose information. You don't know what fluorescent readout there would be in round 2 or in any other round, and this allows you, or you're not able to decode that barcode, so you lose that information. So that's one of the reasons. Another reason is also optical crowding. If you want to really visualize 1,000 genes per cell, it's really hard to identify or image all these individual spots. So one way of doing this or overcoming this is really utilizing super resolution microscopy. So all these dots in a given cell that are really blurry, you cannot make out anything. You can really utilize this technology to pinpoint these spots more accurately. Of course, this is very challenging, highly expensive equipment, very low throughput, so it's not scalable. So another way of approaching this is using expansion microscopy. So again, if you have an image or a cell, let's say, with a lot of these fluorescent dots that you cannot decipher because they're too close to each other, you can actually expand your tissue with a linear expansion protocol, where you then have those two individual dots that you can actually distinguish because they're just further apart. This also helps with reducing the fluorescent background in certain tissues. And this was actually also explored by the people that have developed SecFISH. So they developed SecFISH expansion using this methodology. Of course, showing in this figure, a major drawback here is that your imaging time just goes through the roof. So even for the same region of interest, your imaging times go way up. So this reduces your scalability as well, your throughput. A very elegant way of really approaching the higher multiplexing capacity while still allowing you to deal with optical crowding is how you design your barcodes. So in the earlier examples, I gave you an example of dense barcodes, where in each imaging cycle you have a fluorescent signal for each transcript. And of course, this can get very crowded very fast, but with sparse barcoding, you also include the absence of signal as informative for your barcode. And this in effect dilutes your number of fluorophores per imaging cycle dramatically. And this allows you to interrogate much more signals and thus transcripts in a given sample. And the two major landmark technologies that utilize this approach are called MERFISH and SecFISH Plus. So these slides are a bit complicated perhaps, but generally how it actually works is you have all your transcripts, you hybridize them with target probes, and these target probes have two different tails with different positions on which you can actually bind or hybridize these encoding probes. These encoding probes have then different fluorophores, and this allows you to really build your barcode. And in every imaging cycle, not every transcript has an encoding probe that has a fluorophore on it. So it really has a sparse barcoding built into that. It also has the error correction. So what I explained earlier about the mishybridizations, you also have error correction in here, where if you have one change by any artifact, one change, for example, does not mean that the barcode is going from gene A, let's say, to really gene B. So there is room for errors, and we can also use these chemistries to correct for that. So these technologies have been demonstrated to really interrogate almost 10,000 genes per sample, so very high multiplex capacity. Of course, these detection efficiencies, they range between 80–90% compared to single molecule fish, just because of also optical crowding and these type of things. But one important thing I want to mention here is these technologies are often destructive, in a sense that you need tissue clearing. You need to remove the proteins from your tissue to be able to actually use these protocols. So the next category is called in situ sequencing, in which you actually use padlock probes targeted against a certain region of the transcript. And what these technologies have in common is they use these padlock probes, but also they use an amplification method through rolling circle amplification, in which they create these nanoballs inside the tissue. And with other probes targeted against these nanoballs, you can actually have a lot of fluorescent signal to detect the location of these nanoballs. So it's a very powerful technology in terms of multiplexing. However, there are some downsides here. You have, of course, this enzymatic amplification step, which reduces the sensitivity, the capture efficiency. It's way less than single molecule fish, and also the resolution, because you're generating these nanoballs. These are actually quite large dots. However, it allows you to have a big field of view, like you see in the images here of the mouse brain, and the use of padlock probes has the advantage to really detect SNPs, for example. And the last category is called the spatial sequencing approach, where I think the most famous example here is from 10x Genomics with the Visium slide, where you have, in this case, a slide, a carrier with spatially barcoded oligos that have a poly-DT end, and you basically place your tissue section on top of this array. You permeabilize the tissue, and these transcripts, they float down and they bind to the poly-DT oligo, and then you generate a library for sequencing. So this is not a imaging-based technology, but sequencing-based, and this also increases the throughput. So in this space, you actually have many other competing technologies. For example, you have StereoSeq with resolution in the nanometer scale, very big field of view. You also have SlideSeq, which uses actually beads, a bead array with also these poly-DT oligos attached to that to capture the RNA. And a very recent publication also displayed the SEQ-scope approach, which, as opposed to StereoSeq, which is compatible with BGI sequencing, the SEQ-scope actually has the same principle, but then with the sequencing by synthesis methodology used in Illumina sequencers. So basically, you use flow cells to actually capture these transcripts in your tissue. So very interesting things going on in this region of spatial transcriptomics space. The big strength here is the unbiased capture of mRNA. So it's really poly-A based. However, it's not truly single cell, because you're dealing with an array. You cannot really delineate a cell and say, okay, these transcripts are only in this cell. And there is a massive trade-off between resolution and sensitivity. So the smaller these capture areas, the less transcripts, of course, they can capture. And you're targeting highly expressed or mid-expressed genes, because you quickly saturate the capture rate of these beads or these arrays. However, the throughput is there, so very large field of view, and it's sequencing based, so that's a nice advantage. So really, to sum up all the things I mentioned today, if we look at the major parameters that are important in spatial technologies, based on the categories that I listed, we see that it really depends on the applications you want to use a technology for. So do you want to go for highly multiplexed targeted technologies with a high resolution? I think barcoded FISH approaches are very powerful and one of the best in the space. However, if you really want to have much more throughput, really have an untargeted view in your tissue, I think the spatial sequencing methodologies are a better fit. But it's important as a user to ask the right questions when dealing with a certain technology to really know which technology is right for you. And I'll hand over again to Jasper to explain more about what's up and coming in the field of spatial transcriptomics.
So now that we've seen that spatial transcriptomics have come a long way, it is time for us to look at what is next for the field. And here we want to highlight three main points: increasing the resolution, the dimensionality, and the integration of data from other omics spaces. So let's start with the resolution. Even though some imaging-based techniques have been able to provide subcellular resolution, this is not sufficient to solve all biological questions that we have in transcriptomics. And I want to use this picture of an interneuron displayed here in the middle to highlight the two major challenges that we need to overcome. First, the density of the transcripts within the cells. And here we are looking at 322 somatostatin transcript at subcellular resolution. And I think from this, it is easily feasible that if there are many more, molecular crowding of signals might become a problem, and individual transcripts can no longer be distinguished from one another. So this is something that we have to keep in mind. And the second problem that I want to highlight is the resolution itself. So even though these pictures are already quite impressive, the resolution is still not enough to resolve expression in small subcellular structures, such as synapses. And to visualize transcription at synapses, we would therefore need to find ways to increase our resolution. And there have been some very promising techniques to do so that Jeroen also mentioned in his part of his talk. For example, the use of expansion microscopy has been used to lift structures above the diffraction limit, or super resolution microscopy has been used to image below the diffraction limit. However, each of these techniques come with their own challenges and complications. Now that said, further increasing the resolution of spatial transcriptomics will be a challenge that we need to address in the future. The next challenge for spatial transcriptomics that I want to highlight is the dimensionality. And one of the major problems we are currently facing in the field is the fact that most of the technologies we have introduced are working on sections. And these sections, due to the nature, have a very limited depth, which, as you will see in a second, might become a problem. So here I show an unpublished Alzheimer's studies that we did in our lab to investigate the changes in cellular composition in relation to the distance to an amyloid plaque. So in the image on the very left, you can see the antibody stained plaque in yellow and all the nuclei stained in blue. And we then measured the distance of the nuclei to the plaque by drawing concentric circles around the plaque. And then we used our transcriptomics data to determine the cell types that are closest to it. And our data here shows an enrichment of microglia close to the plaque, while the presence of excitatory neurons was reduced, which is a nice finding. However, though we still think that this data is of really high quality, upon analysis we asked ourselves how much of a problem it really is that we lost all the information in the z-plane and have no way to determine whether the cellular distance to a plaque in xy direction is also true in z. So in order to be sure about our findings, we would need a thicker piece of tissue and overcome all the challenges that come with it. Now, when you think of this challenge, the two most obvious solutions that come to mind would be either to work on multiple slices and stitch these together, which would require algorithms and procedures that are capable of doing this without much information loss in the slicing plane. Or, on the other hand, one could work with thicker sections and use imaging techniques that allow imaging in deeper sections of tissue, such as confocal imaging. However, here it is important to note that even if the imaging challenge can be overcome, reliably diffusing probes into and out of a thick tissue block might become a challenge in itself. Now that said, by doing research for this presentation, I stumbled across studies that have succeeded to do whole mount in situ hybridizations. And here I present an example from a protocol published in 2013 that used zebrafish embryos for in situ hybridizations. However, such experiments also show that even though it is desirable to work on intact tissue, these technologies still have a long way to go to yield comparable data to the currently used methods in spatial transcriptomics. And then, last but not least, we cannot regard transcriptomics as a field in itself. We know that the transcriptome is highly connected not only to the genome, but it's also the epigenome that plays a crucial part in regulating gene expression. Several studies have shown that combinations of transcriptomic and epigenomic information, such as a combination of, for example, single-cell RNA-seq with ATAC-seq and ChIP-seq, have increased our understanding of the transcriptome significantly, for instance, by facilitating our marker gene selection for individual cell types. And along the same lines, having a profound understanding on the transcriptome is crucial for a good understanding of biological processes. However, today we know that RNAs can travel long distances within tissues, so the location of a transcript must not necessarily be its origin. And to make things even more complicated, we also know today that simply knowing the position of a transcript does not always correlate with the position of the corresponding protein. So a combination of knowledge between spatial proteomics and spatial transcriptomics is therefore inevitable. And of course, it doesn't stop there. We are convinced that better data integration from all the omics spaces, from genomics to metabolomics, will help us gain a holistic view of biological processes and boost our understanding of life itself.
So thank you everyone for tuning in. So if you have any questions, feel free to post them now or just send us an email. If you want to know more about what we are doing at Resolve Biosciences, what type of spatial transcriptomics technology we are developing, where do we fit in this space, etc. And if you want to start a project with us, just feel free to email us. And thank you very much for attending this webinar.